Hey everybody, Mr. Murphy here. I'm going to go through 5.2, which is all about diffusion. So this section is pretty full of vocabulary which will be valuable to know and to understand. If this is like something where Quizlet or flashcards or things like that can help, I think that's a good thing because you don't want to have to stop and think through the entire process when we say things like isotonic or hypertonic or things like that. We want to become familiar enough to understand it kind of quickly. So just be aware of that. Otherwise, I think this section is primarily review, or hopefully, and should hopefully come back to us pretty quickly from earlier biology classes. So in the previous section, we talked about the membrane, which is selectively permeable, and it's permeable by some things, but not others. That's what that means. And it is because of that hydrophilic, hydrophobic, hydrophilic double layer membrane that's possible. And the fact that the cell membrane is peppered with all these different proteins that can facilitate the movement of things across that membrane, which allows the cell to not just be selectively permeable, as in only some things can get across, but also it can be selectively permeable in the sense of like it's choosing what can go across at different times based upon the environment or based upon the homeostatic needs of the cell. So there's two main types, passive transport and active transport. In this section, we're looking at different examples of passive transport. So everything that follows in this video will be about passive transport, which does not specifically require energy in order to happen. And it is entirely based upon the concentration gradient. So things moving down their concentration gradient from a high concentration to a low concentration. So this can happen in two different ways. Simple diffusion, which is just the movement, the simple movement of things through the phospholipid bilayer, sneaking between those phospholipids, traveling through the hydrophobic region and into the cell, versus facilitated diffusion, which needs the help of one or more proteins. So diffusion is just the process of things to move towards equilibrium. It's random, it's chaotic, it's just based off of molecules bouncing into each other. You know, you spray a whole bunch of Lysol in one corner of the room, it's going to disperse throughout the rest of the room simply because those molecules are bouncing into each other and sending each other off in different directions, not to mention interacting with the air molecules and all sorts of things. And in the end, just that randomness is going to lead to dispersion and an equilibrium being established in whatever the confines of that container happens to be. So this can depend on a number of factors. First off, the diameter or actual size of those molecules. Smaller things diffuse faster, which should kind of be logical, right? Larger molecules are going to require more energy for movement, and it's going to be slower process for them to disperse or certainly to cross a membrane, as we'll see. The second thing is temperature. Temperature is really ultimately just a measure of kinetic energy of molecules, and so the higher the temperature, the faster the molecules, the faster it's going to spread. Right? So, you know, you could have a tray of some delicious food sitting there, but if it's all cold and frozen, it's not really going to emanate its smell very widely or very quickly. Meanwhile, you know, freshly cooked chicken tenders or something are going to make a whole room smell deliciously pretty quickly, just because of the temperature of those molecules. They're moving much faster and able to spread much more quickly. Concentration gradient refers to the difference in concentration from one area to another. The higher the concentration in one area, the faster you're going to see movement towards another area of molecules, simply because that much larger concentration is going to be facilitating much faster change. When we talk about simple diffusion occurring straight through the phospholipid membrane, a molecule that's hydrophobic is going to be able to sneak through that fatty acid layer of the the lipid bilayer. The polar molecules not gonna fly so much because they're not gonna have any. There's going to be much more of an attractive force on the hydrophilic sides that's going to discourage their movement straight through the membrane. If a concentration is high enough and if there's enough temperature or a high enough temperature, yeah, it's possible for polar molecules to sneak through, but again, that's not going to be their primary mechanism. Osmosis is the movement of water specifically. So water is a polar substance, and it is pretty small. And so water can pass through the membrane if the concentration really drives it, if there's enough of a concentration difference. But primarily water is going to be traveling through special membrane channels, which we'll talk about shortly. So when we are looking at any two solutions that are separated by a membrane, these are the terms which hopefully we're vaguely familiar with, and we want to make sure we have confidence in using. A hypertonic solution is one that has a higher solute concentration. What is that solute? It could be salt, it could be sucrose, sugar, it could be any number of things, but the point is that it is a higher concentration solution that we refer to as hypertonic. Isotonic solutions are when both sides of the membrane have the same concentration, and then a hypotonic solution is going to be the one that has a lower solute concentration, which means that if you have a hypotonic solution on one side, you have a comparatively hypertonic solution on the other side until movement is allowed to occur, at which point you'll end and reach equilibrium when they are both isotonic to one another. In that circumstance, it doesn't mean that movement has stopped. It just means that on average, any movement one direction is going to be countered by the same amount of movement in the opposite direction, and that equilibrium will be maintained. So here's some images that demonstrate this. Here we can see a much higher solute concentration just by the number of little dots here. That's the different solute molecules, and we see that the water is passing through this selective membrane. Apparently the little dots are too big, they can't fit through, but the water can, and so the water is going to move out of the cell, in this case on the left side, and it's going to move into that extracellular area towards the high concentration of solute. In blood cells, this is when our cells become dehydrated. So when you are actually dehydrated, you haven't been drinking enough water, that becomes dangerous because it can cause a misshapen characteristic to your cells as the water is physically drawn out of them. So here we can see a red blood cell, which is usually this nice little dimpled disc shape, and here it is becoming malformed by the loss of water. That change in shape means it can't interact as well with oxygen and various other molecules, which is going to hinder its ability to do its job. In plant cells, we see this where the cell membrane actually shrinks down. And so normally you can't really discern a cell membrane from the cell wall. The cell wall is extracellular, but the cell membrane's bumped up against it, typically in a plant cell. When that plant cell is starved of water, you can see how the inside sort of deflates. And this is what causes the squishiness that leads to wilting in plants. An isotonic solution, and here's that nice plant cell where, again, you can't really see the cell membrane versus the cell wall because it's nice and filled up. Here's a nice blood cell, and you can see equal concentrations of solute on both sides. It's the reason why somebody's dehydrated. When somebody's dehydrated or goes to the hospital and for any reason needs an IV bag, saline is used instead of pure water or something that contains tons of salts. And saline is just the right concentration of solutes in order to match what is ideal for the human body. And so the goal there is to replenish fluids, yes, but do so in a way that's not going to shift the tonicity of either side, outside or inside of the cells. Hypotonic can be actually just as problematic in humans in that having excess amounts of water or an increased amount of solute within the cell can lead to water diffusing through osmosis into the cell, which can cause the cell, here's another blood cell, where it's actually swollen and become malformed in sort of the opposite direction, which can actually be dangerous. People have died from excessive water consumption. You don't have to worry about that. Like you went to the water fountain 3 times instead of 2 times in a day. Oh no, it's not like that. But pushing the limits of your body, it can actually become dangerous. Or if you were drinking like pure, distilled water that doesn't contain anything except H2O, can throw off the salt balance outside of your cell versus the inside, which can drive water to move excessively into your cells. And here you can see, again, with the plant cell, it's hard to discern what's going on here, but these plant cells are causing extreme pressure against the cell wall, which can be problematic. That's why you can kill a plant by overwatering it, just as much as underwatering. So the concentration of solids determines the direction. This is important. direction of osmosis can go either way. That's sort of the point of the preceding cells and examples, that water can leave just as easily as it can come into the cell. In the case of plants, that's utilized as a functional benefit, that the plant cell takes up water into its vacuole to maintain basically an inflated interior membrane to push against the cell wall in order to provide the pressure needed for that cell to withstand the forces of like the entire rest of the stem or trunk or whatever of the tree, that when those cells are compressed by the weight of the plant, it's able to withstand that pressure. That's why if you take a flower, you forget to water it, it wilts. You water it the next day, hopefully before it's dead, and it'll come right back up. The force is created by the pressure within the cell by the uptake of water. So diffusion can happen with some additional help, and that can happen in two primary ways. Channel proteins, which are basically, like the name implies, just little channels through the cell membrane. This would be a transmembrane protein existing between both sides of the lipid bilayer, and that's allowing for the passing of certain molecules specific to the shape of the pore there that they'll be able to pass through. Now, it's bidirectional, so whatever the direction of diffusion happens to be, that's going to be the direction, whatever the direction of the concentration gradient is, that's going to be the direction of the diffusion. Likewise, carrier proteins are very similar in that these are proteins which grab onto a molecule and then, not grab onto, but because of a chemical attraction, they match up, and that causes a change in the shape of the protein, which then delivers that molecule across the membrane and into the cell. It's a subtle difference, but an important one, just to understand that there are different types of proteins to facilitate transport in different ways. Some are just more straight paths that molecules can get through, and others require sort of a conformational change. In both cases, we call this facilitated diffusion, and like the name implies, to facilitate something is to help, so it's proteins helping the process of diffusion. It's still movement from high to low concentration. Ion channels can be a little bit different. Some can actually be gated, and these are channels specifically for ions. So like sodium ions, Na+, or Cl- or Ca2+, these different ions which are prevalent inside of our cells, outside of our cells. Potassium's another one. Basically all the things that you see like advertised on the side of a bottle of Gatorade, the electrolytes, those are our different ions which our body needs for muscle control, for all of our neurons that are firing. Just a variety of ions are needed for transmitting signals into and out of cells. They can be gated, which means that we can control the allowance of ions to diffuse in or out of the cell, which is a huge component in the mechanisms by which the nervous system works, in that by creating a control mechanism to allow certain things in at certain times because of different stimuli, that allows us to detect changes. So for instance, like when you touch something, you can feel that because that physical pressure is allowing certain gated channels to open. Other channels can be controlled by electrochemical balance, so that if a whole bunch of ions in one region of the cell enters into the cell and makes the cell positive, that can then open a voltage-gated channel, which allows other things to move in or out and basically propagate the signal. Any stimulus that's like a chemical signal that's landing on the cell and opening or changing one of these proteins is called a ligand, which is, again, just a fancy name for the signal that's been received and caused a change in a protein. So a ligand-gated channel would be one that when that ligand lands, it changes something about that channel, and it therefore leads to different action occurring. Voltage-gated ones open or close in response. So that's what I was mentioning before. So here's an example of one of these channel proteins, and here you can see it's closed. So this is a cool benefit of using these proteins in this way, that because proteins are so specific to shape, their functionality can sort of be activated or deactivated by creating the condition that allows for the particular shape that lets the thing happen. And so here we have a closed channel which waits for this little signal molecule to land here. The protein doesn't have thoughts. It's not waiting, like looking out for the ligand to come landing. But what's happening is when this little chemical molecule lands here, it changes something about the shape of this protein. It tugs and it pulls on its amino acids in such a way that the shape change correlates to now the ability of this carrier protein to allow molecules to pass through it. And so here you can see a high concentration of this little red molecule here. The ligand comes, signals the cell, yep, time to open the channel protein, and now diffusion's allowed to happen from high concentration to low concentration. There's nothing that says it needs to be in this direction. It could be in either direction. It just depends on what the conditions are. So sometimes the cell is maintaining a high concentration on the inside. Sometimes the cell is waiting for a particular stimulus to allow high concentration to move into the cell. Water crosses membranes faster than just simple diffusion because there's a few ways in which it can get in. It can hitchhike with ions such as Na+ as they pass through channels. And aquaporins are specific channels that allow for the movement of water into or out of the cell. And kind of like the name implies it's aquaporins. These are water pores in the cell that allow for very fast movement of water into or out of the cell. Water is such an important molecule and such a foundational part of homeostasis that the presence of aquaporins is significant in a large part of, for instance, the human cells. Here's an example, which is kind of cool, demonstrating the effectiveness of aquaporin, where aquaporin mRNA is injected into the cell. That mRNA then grabs onto a ribosome and produces proteins and the whole process with ER and the Golgi and all of that, which ultimately leads to the production of aquaporin channels, the proteins that allow for the movement of water. And so the prevalence of these aquaporins increases after the cell's been injected with this mRNA, which then allows for the movement of water. And so you can see in these results that little oocytes that were injected with aquaporin mRNA produce more aquaporins, and suddenly their relative volume skyrockets compared to the cells that didn't have the mRNA coding for aquaporins. So again, demonstrating the effectiveness of, or actually demonstrating the functionality of these aquaporins in the ability to transport water across the cell membrane. Carrier proteins do this by binding their substances. So glucose transporters are great examples of carrier proteins, and we'll see in the next slide a visual of this. But the glucose binds onto the carrier protein, changes the protein shape, which then basically drives the glucose into the cell, and you can see how we represent that here. Obviously, on a cellular and molecular level, proteins don't look like nice little regular blobs, but they're actually tangled messes of amino acids. But still, this is a good representation of ultimately what's happening, that the protein is grabbing onto the glucose, that glucose has a particular shape and different hydrogen bonding sites and things like that, which changes the shape of the protein, which then allows for the glucose to be released on the other side. So it's kind of like one of those turnstile revolving doors that some places have, where it's allowing you to go through, but you kind of got to go through a process. You got to enter in, you got to go in the right place at the right time, and then you get spat out on the other side. It's important to know that any example of facilitated diffusion has rate limits, because even if we have a huge concentration of glucose outside of the cell, which would be as we move to the right on this chart, at a certain point, the rate of diffusion is going to stop going up because, okay, you've got so much glucose, there's plenty of glucose, but there's simply not enough carrier proteins to keep up with the demand. And so even if you keep introducing more and more glucose, you're not going to get faster and faster diffusion, just because literally all of those proteins, those carrier proteins, are letting a glucose in, changing its shape, letting it in, grabbing a glucose, changing its shape, letting it in, grabbing a glucose, changing its shape, letting it in. It's just that a process is happening as fast as it possibly can. So there are limits to facilitated diffusion in terms of the rate in which it can happen. So that's called saturation, and this slide's just discussing what I just explained. That's the end of simple diffusion and facilitated diffusion, both examples of passive diffusion, which does not require energy. In the next section, we will discuss active transport, which does require energy. As always, if you have any questions, let me know.